Hi, everyone. We're going to get started. I am going to give a brief introduction for our speaker today, Dr. Gostel. He's here visiting us he's currently at the Research Institute of Texas, or the Botanical Research Institute of Texas, BRIT. And he's been working there since, I believe, 2018. He's a research botanist, and then he graduated in 2015 with his PhD in environmental science and policy. Sorry, it's a unique PhD, so I had to write that one down. He's researching currently in plant systematics and evolution, and while also being the director of the Global Genome Institute Gardens. So the GGI Gardens is what they acronymed it as, where he's also the director and is working internationally collecting and trying to preserve plant biodiversity in biorepositories. And to safeguard genetic resources for plants all across the world. And I know from a little bit talking to him, he seems to have traveled all over, and that's really awesome. And I'm excited to hear from him today. So let's give him a warm welcome.
All right, let me just mount this microphone thing. Hopefully that is good. All right. So thank you everybody for being here. Thanks especially Trish for the invitation to come and speak a little bit, talk to you about some of my research, or not really research so much, but an outreach program that I've been involved with as program director and most recently director since 2015, since the program was started. This is my first time here at Texas A&M. I've had a great visit so far, thanks to the department for being a really gracious host. Learned a lot, and I've had a really good time talking to students and faculty here. So as Cassie mentioned, I'm a research botanist at the Botanical Research Institute of Texas, and actually I have neglected to include the Fort Worth Botanical Garden on that. I apologize. For the past year, the Fort Worth Botanical Garden and BRIT have been under the same management as part of the Botanical Research Institute of Texas. So I'm really excited to talk to a horticulture department because my background is in plant systematics, as Cassie also mentioned. I have worked with plant evolution and mostly phylogenetics for all of my career. And to speak with a horticultural department is really exciting for me because for the past 6 years, I have led a program that is focused on working with botanic gardens, or as I like to say, building bridges between the horticultural and research communities by sort of combining traditional collections-based research with genomics, utilizing innovative approaches to teamwork. So just to give a brief outline of what I'm going to talk about today, I'm going to start with just a little bit of an introduction to some of the changes that have motivated this work in the 21st century, some of the changes that have really impacted living collections in an important way. And this focuses on the collections themselves, the field of genomics, as well as the tools that have allowed for new approaches to collaboration and coalition building. I'm then going to talk about this broad area of my talk today in a more narrow context of developing a model of approaches to building bridges through the Global Genome Initiative for Gardens program. So I'm going to talk about some of the data that went into building that and what it took resource-wise to actually make that model work, and what the impact so far has been. I'm then going to wrap up with a little bit of discussion of the future directions we have for the program, what comes next after we have built a massive collection of genomic resources for plants across the plant tree of life, and then how we're focusing on maintaining bonds and building infrastructure to support the bonds and the partnerships that we have built. So I like to characterize this introduction to the talk about living collections in the 21st century, really focusing on some of the evolving techniques that have led to new paradigms for living collections, as I mentioned. This includes our increasing awareness of the biodiversity crisis as a result of myriad things: population growth, climate change, the way that gardens have really adjusted in many ways their mission to focus more and more increasingly on research and conservation. Also, of course, the genomic revolution has completely transformed the way that we understand biological diversity, the way that we study biological diversity. And then third, I think one of the, of course, paradigm shifts has been promoted by the advances that we have seen in computational science and digitization that have allowed for what I like to call a leveling of the research playing field. Whereas previously, a lot of high-level research resources were locked in a small number of institutions that were well-funded and had lots of access to a number of resources. And what computational science and digitization has done has allowed someone across the world to access and utilize data or collections and see them and be involved with them in a way that they never maybe could have otherwise had an opportunity to do. So when I talk about collections, I like to talk about them in a two-dimensional matrix. There, I think, are really four ways to characterize collections. I think about them first in terms of how we preserve them, whether we preserve them in a cold or a warm condition. And so I'll give you an example of these. And then the other side of this matrix is whether those collections are living or dead. So as an example of cold and warm, living and dead collections, I present to you as an example of cold, dead collections, what is often called a biobank or a biorepository. And this is what I'm going to be talking about a lot in my talk today because it's a focus of the GGI Gardens program. These are facilities that are dedicated to storing, preserving, organizing collections that are utilized for genetic research, genomic research. They can include frozen blood samples from birds if you're studying pathogens. They can include what I've been spending the past 6 years collecting, which are vegetative leaf samples that are preserved often in liquid nitrogen or dried on silica gel and frozen at either -20 or -80 in a freezer so that they can be preserved in an ideally pristine condition for later genomic research. On the other hand of the dead collection situation, we have warm collections. These are things like herbaria or your traditional natural history museum collection, where the collection is not necessarily warm. Hopefully it's not too warm, but it's certainly not kept in a refrigerated environment. So herbaria or a paleontology collection of bones that is kept at ambient room temperature at a relative stable humidity, things like this. Now, on the other hand, living collections can either be cold or warm as well. And so a great example of a cold living collection, of course, are germplasm collections. Very often, I think of things like the USDA germplasm collections or the Svalbard Seed Bank or the Millennium Seed Bank that is sponsored by Q. And then, of course, the focus of this talk really is going to be on the warm living collections. And in this case, I'm talking about plant collections in botanic gardens or arboreta or what have you. In that same vein, of course, warm living collections might also include aquaria or zoological parks, right? So anyway, one of the things about warm living collections, especially in the 21st century, is the realization that they've been severely underutilized for research purposes. And this is captured by a recent paper by Michael Dozman, who's the keeper of the collections at Arnold Arboretum. He sort of published this call to action for the Botanic Garden community in 2007. And in it, he highlighted a couple of priorities that could be leveraged by Botanic Gardens and the user community of Botanic Gardens to avoid what he called this collections crisis. What happens if all of a sudden we lose a collection at a Botanic Garden? We saw this recently here in Texas. Hurricane Harvey caused, luckily, not as catastrophic damage to, for example, Mercer Botanic Gardens in Humble, Texas, as could have happened, but certainly could have been a lot more disastrous than that. With climate change, we see the incidence of catastrophic weather events that could severely impact these collections. So that's what he means by the collections crisis. So the first recommendation is build research connections between the providers, in this case, the garden, and the users. So build and enhance connections that exist between those two communities. Second is expand collaboration, not between the provider and the user, but in this case, within the garden personnel itself. There is certainly a tendency for any of us who work in a particular part of our job to isolate or focus on the aspects of our job that are mission critical. But reaching sometimes across department is challenging, it's time consuming, it takes resources, and it detracts from what we've been hired to do. But it is really important, especially if we are to promote research activities at Botanic Gardens. So what Michael means by garden researcher collaboration and garden, for example, at Fort Worth Botanic Garden and the Botanical Research Institute of Texas, we work in separate buildings as research botanists from the horticultural team at the garden. We very rarely interact. When we do, it's usually for a team building event, but it's not very often, and not often enough for sure, in a way that talks about how we can mutually collaborate for the benefit of the garden, for the institution where we work. And that's unfortunate. The third recommendation by Dr. Dozman was encouraging gardens to more effectively manage information. This is very common in many Botanic Gardens. Every single one where I've worked does not have, I've never seen a perfect living collection database. And that means a database that records information about when a plant was collected, all of its provenance, information, where it can currently be found in the garden, whether or not it died 10 years ago and nobody took notice and recorded it in the database. Not just this, but also integrating data sets across an institution from the living collection database, helping that database communicate with other databases. If a Botanic Garden also has an herbarium, is the living collection database communicating with the herbarium database? Have any of the collections in the living collection been vouchered and deposited into the dead warm collection, the herbarium itself? And then if someone has ever come to visit the garden for a research purpose, collected something, published their findings that were supported by that garden, did the garden follow up, or did the researcher who came and used that material, did they follow up with the garden and let them know that, hey, we published, thanks to your partnership, thank you for letting us use the collection. Just wanted to let you know, please share this with your board so that you know the impact of your garden is really helpful. It's facilitated our research in an important way. So that's sort of my soapbox about collections in the 21st century. The second part of the talk, of course, is about genomics. Why genomics? Well, of course, this is integral to understanding biodiversity in the 21st century and the evolution of biodiversity in the 21st century. Genomics is the wonderful silver bullet to solve all of our research problems, right? So we hope. And with that in mind, everyone has been, over the past 10, 20 years, gobbling after developing new approaches to utilize genomic data. And it's been wonderfully successful. Technology has advanced to such a point where the cost of sequencing is actually comparatively quite cheap. And I was struck in 2015 when I started developing the GGI Gardens program to realize that the limiting factor for very large, ambitious sequencing projects is not the cost of sequencing anymore. It's actually getting access to tissues of the species on Earth. If you were to, for example, say, I'm going to sequence the genome of all species in Texas, how would you go about doing that? Well, the sequencing part is pretty easy, right? You have a core facility, you have technicians who are going to sequence the data. How are you going to get all those samples collected and run through your pipeline? That's the challenge. This is reflected, I think, really illustratively by a paper published by Rod Page, who's an entomologist in Edinburgh in 2016. He coined this phrase dark taxa, which what he means by that is these are operational taxonomic units. They're taxa. They are populating ecological and molecular databases, but they don't have an actual taxonomic name associated with them. And so this is a plot of sequence data in GenBank of invertebrates from 1995 to 2015 over a 20-year span. In blue, you have sequence data in GenBank that has a taxonomic name associated with that sequence. And in red, you have sequence data that does not have a taxonomic name associated with it. And so you see this really troubling, kind of scary trend as a taxonomist. I'm going, ah! And we're seeing this massive spike in sequence data that does not seem to have any taxonomy associated with it. And this is partly driven by the advancement of high throughput sequencing technology. Part of it is, of course, new methods that allow us to do things like metagenomics or metabarcoding, where we can gobble up massive amounts of samples and sequence them and spit them out, deposit them in GenBank. But we don't have enough taxonomists in the world to name them or figure out what we gobbled up and sequenced and spat out. It's not necessarily so scary, but it does draw a lot of attention to the taxonomic impediment. The fact that we need to be training more taxonomists to help us understand what this data actually is. So in response to this taxonomic impediment, especially as it's been manifested with sequence data, biodiversity genomics, and sorry, the top of the slides is kind of being cut off, is a field that has been promoted as a way to sort of unite the collections base with the genomics base and to help us come together using genome scale data to both do things like achieve conservation targets, but then just generally understand biodiversity. And some of these projects you might be familiar with without realizing it include things like the International Barcode of Life project. This is a biodiversity genomic project. It falls under that sort of paradigm. Another is the Q Plants and Fungal Trees of Life project. Another is the Earth Biogenome project. This was announced in 2018 as an international effort that seeks to sequence the whole genome of all eukaryotes on Earth in 10 years. Another, of course, you may be familiar with is the 1,000 Plant Transcriptome Project or the 10,000 Plant Genome Project that have been funded by the China National Gene Bank and Beijing Genome Institute. So these are some really cool, high-level collaborative projects that are multinational. And they're focused on trying to tackle the big sequencing and genomics questions of our days. So that brings me to the sort of third part of the talk, point of the talk, which is about teamwork and bridging gaps within and among institutions. So I do want to say that using and teamwork at using living collections and at Botanic Gardens is not a novel concept. In fact, there are numerous, I can rattle off many, the Global Crop Trust, the Millennium Seed Bank with Q, ArbNet, the Arboretum Net that's managed by Morton Arboretum, which has established an accreditation scheme that can help encourage gardens and arboreta to incorporate more research and conservation activities into their organization's mission. The American Public Gardens Association, in collaboration with the US Forest Service, funds a tree gene conservation project around the US. The USDA, of course, is just obviously smack dab in the middle of many of these projects. Others include the work that Botanic Gardens have done through the World Flora Online to promote an online digital flora of all plants on Earth, to consolidate taxonomic information, to share it with gardens, with the users of gardens and their data. Botanic Gardens Conservation International, which funds many conservation projects around the world, Center for Plant Conservation, and things like the Global Conservation Consortia for various plant lineages. So I don't need to keep going on this. Another one of those, and the one that I'm going to talk about for the rest of the talk today, is one that was the brainchild of my postdoctoral advisor, the late Dr. Vicki Funk, just an incredible person. I'll talk about her in a minute. But I had the great fortune to work with her to build this facet of a larger initiative called the Global Genome Initiative. This is a Smithsonian-led project that was funded in 2008, $15 million, with the mission of preserving and understanding Earth's genomic biodiversity. So what their goal is to strategically mobilize a genome quality tissue sampling effort for the entire tree of life, not just plants, not just animals, everything. And the purpose of this was to illuminate those dark taxa on the tree of life. So where GGI Gardens comes into this is a funny story. I'll go really quickly, but Vicki Funk worked very closely with the director of GGI, John Coddington, who was a curator of spiders at the National Museum of Natural History. When he assumed directorship of GGI, she was always thinking big. She had an incredible way of bringing people together, bringing people from different departments together, sitting them down and getting them to work together, collaborate. She was also very gregarious and fun. And she saw this as an opportunity to direct a lot of funding to support and encourage research collections from botanic gardens around the world. And she nor I had any idea what we were getting into at the time. And it actually started as a bet. She was sitting with John at a happy hour one night and said, "John, GGI, your mission is to collect as much diversity on Earth as possible and preserve it to enable whole genome sequencing projects." Yeah. Okay, well, I bet you a bottle of wine that you can find at least half of the genera of plants on Earth in botanic gardens. And that's really convenient because it's easy to sample from a botanic garden. They very often have a well-organized collection. The collection is alive, so you don't have to worry about things like DNA fragmentation, degradation that happens in those warm, dead collections pretty quickly. So you're able to get very high quality tissue that's nearby very quickly. He said, "I'll take you up on that bet." So we started. There was a pilot project in January 2015 at the US Botanic Garden. I joined as a postdoctoral fellow in August 2015 as program manager. And we sat down and we said, okay, we need to set some targets. First of all, how many genera do you think, what do you think we're going to collect, Vicki, from botanic gardens? Because I had no idea. I came from museums, from working in herbaria, and I had worked with a lot of botanic gardens throughout my career, but this was new. And we decided, well, I bet we can find all families of plants on Earth in botanic gardens. These are total estimates. We had no really formal information to help guide these estimates. We said, I bet we can get 50%, half the genera. That's what the bet was staked on. Half the genera of plants in botanic gardens. and then about a quarter of species of plants in Botanic Gardens. And then we use that as a guide to set some targets, some goals for the program. And we said, okay, well, before we get started, in phase 1, let's say we want to sample and preserve at least one species from each family of plants, from 50% of genera, and all species as this wildly ambitious moonshot for the project. So in 2015, we're sitting down, we're thinking about how we're going to start this. I'm starting an intern program where I'm going to the National Arboretum, the US Botanic Gardens, Smithsonian Gardens, to go out and collect. And we're also doing a number of things, working with other large organizations to figure out, okay, how are we actually going to make this work? Because we've been entrusted with this responsibility, and now we got to do it. So first of all, we started working with Botanic Gardens Conservation International very closely because they organize and operate a website called Plant Search. And Plant Search is a database. You can go online, you can enter a taxonomic name, and it will return to you a list of Botanic Gardens where you can find that in their living collection. So BGCI is the keeper of records for global Botanic Garden diversity. We talked to Paul Smith, who coincidentally was working with Ross Mounts and Sam Brockington at Cambridge University on estimating, trying to figure out an answer to this very question. How much diversity is in the world's Botanic Gardens? So in 2017, they published this in Nature Plants. It talks about the ex situ conservation value of Botanic Gardens worldwide. So on this figure in the top, you see a red dot on the world map where a Botanic Garden in BGCI's database is found. In the bottom panel is sort of a heat map that shows circles associated with the species diversity of those Botanic Gardens according to the inventories that BGCI has in their database. As you can see, there's a massive disparity in the global south and between the global south and global north. This is not because obviously there's not much diversity there. It is largely due to the fact that BGCI does not have inventories for the gardens from this part of the world. So that's one area that could certainly help figure out where things are. Nevertheless, using the data from only about a third of the world's Botanic Gardens, they found that indeed Botanic Gardens are, and this is tracheophytes, vascular plants, by the way. Botanic Gardens are home to about 30% of all species of plants. Ding, check the box for our estimate of species, that was cool. Almost 60% of all genera, ding, check the box for genera. And for tracheophytes, 75% of all families, and about 93% of all seed plants. So not ding for the 100% of families. But keep in mind, this is only a third of Botanic Gardens in the world. There are another 2,000 Botanic Gardens that aren't even in that data set. So we thought, I think we can probably say if we were to actually have data for those missing gardens, we could get it. Okay, so a big collection program like this requires a very large, both physical and data infrastructure component. And this actually, when GGI started, did not exist. So for about, in the 1990s, an organization called ISBR was developed, was founded, that is a biobank organization that talks about they establish best practices and standards for biobanks to adhere to. And it started because hospitals were beginning to store tissue from biopsies and things that could be used for genetic analysis of human health and disease. And in those biobanks, they were freezing cryogenically, whatever, these samples. And it was a great model for, obviously, the human health side of biological research. This didn't exist for the natural history and biodiversity research community. There wasn't an organization that set best practices or standards or told a natural history museum, what is the best temperature to store a collection that is cryogenically frozen and intended for genomic research? Didn't exist. So in 2014, GGI established the Global Genome Biodiversity Network, GGBN. And so since 2014, it's grown to almost 100 members. These are institutions like the Center for International Barcode of Life project at the University of Guelph, where they are doing massive DNA barcoding projects, and they have a huge repository of tissues. The Missouri Botanical Garden, which has had a DNA bank for over 20 years. You can go on to tropicos.org, you can search for a plant name, and you can see if it's in their DNA bank. That wasn't a very efficient way of searching for a collection online. If you're a researcher, you have to know that Missouri Botanical Garden has this database. You have to know to go to their website. And it's even worse for most other collections like this. For example, New York Botanical Garden, they have an amazing DNA bank, but none of it is in an online or electronic database. So you have to go to a curator and you have to ask them, do you have a specimen of this? I would like to do my PhD research on this group of plants. It was very involved. GGBN seeks to set best practices, but also create a one-stop sort of access portal for data in biobanks for non-human biodiversity. And so it has grown very successfully, very quickly. And it's not only setting the best practices, but it's also doing so in, I think, an ethically aware way that is in compliance with international policy and regulation, like the Nagoya Protocol, which is a massive, has been a massive and scary thing, speaking of using genetic resources for the international biodiversity community. So GGI is slowly building, and we realized, we need to develop some best practices, some standards for how we go about collecting from a living collection. How do we do that? First of all, we realized, well, after 1 year, after 2 years of this pilot project as an internship program in the Washington metropolitan region, collecting at the National Arboretum, the US Botanic Gardens, Smithsonian Gardens, by year 3, we realized, okay, 1, we're getting massive diminishing returns. For every hour this group of 3 people, me and 2 interns, spend collecting in the garden, we come back with fewer and fewer new, not before collected genera or families, which was our focus at the time. So we need to figure out a way that helps us pinpoint, okay, what are the priorities for collection? Because if we try to take this model and ask other gardens to go and sample from their collection, what is to prevent them from spending half of their collecting effort collecting something we've already collected? That would be a little bit inefficient. So we developed a gap analysis tool, which is a way for a botanic garden or any organization to submit a list of taxonomic names, and it will cross-reference that list, maybe a botanic garden inventory, with the GGBN database to determine whether or not that taxon has already been collected and if it's available in a biorepository somewhere in the world. And so it's a very quick and efficient way to parse through a botanic garden's collections to figure out what might or may not be of use. So we partnered with BGCI to utilize their plant search database and their behind the scenes plant inventory tools to use this to great effect. That allowed us to do things like individually approach gardens that have a disproportionately high number of new and unique taxa and say, hey, we want to let you know you have a very high number of high value taxa in your collection. Would you be interested in collecting those for research purposes? And can we support that through an award? I'll get to that in a little bit. Other things we did was just figuring out, okay, what does the collection project look like? What do you need to bring with you? How many people do you need on a team for a collection project like this to be efficient, to be quick, and not break the bank at the same time? And so what we found is that a 3-person team was ideal. Typically you have a record keeper you have an herbarium voucher collector, and then you have a genetic and genomic tissue collector. And that 3-person team is able to cover a lot of ground in a garden very quickly. Of course, materials are really just the standard kind of field botany materials that you might expect, a plant press, and maybe not standard, liquid nitrogen dewer. Funny story about that, being accosted by the US Capitol Police with a liquid nitrogen dewer. I don't advise walking near the Capitol with one of those. And bags of silica gel for leaf tissue desiccation. We've published these. They're available publicly through the GGI website at brit.org or elsewhere if you look on Google Scholar. Just to give you an example of what some of the resources we developed look like. So this is a website, globalgeno.me, make it easy to remember. It's an application. You can either submit a live query. So if you're in the garden and you're looking at a plant and you're like, oh, I didn't know this was here. You could submit it to this gap analysis tool and it'll tell you and assign a priority for collection based on whether or not it is represented in GGBN. So in this case, none of the 3 genera, this is from like 6 years ago, so no longer applicable, but none of these 3 genera were in GGBN. So they are priorities for collection and preservation through GGI gardens. However, you'll notice their priority for collection also is weighted by whether or not that taxon has ever been sequenced and deposited in the GenBank as a general sequence or as one of the plant DNA barcode sequences. So anyway, it's a really quick, handy tool for that. We also developed a curated iNaturalist project, which is very effective for sharing data. So it was easy as a single team in DC to go around and know what we were collecting in real time and see what needed to be still collected. But once we started to scale this up to multiple gardens, it became very complicated very quickly. So iNaturalist gives you a platform where collectors can be documenting their collection in the field, upload it to the cloud in real time, and share that with the entire GGI Gardens community through this curated GGI Gardens project. And I really encourage any garden to think about making their own iNaturalist project because it allows visitors to the garden to document what they see in your garden. It gives them a kind of extra special way to interact with the garden. And it gives you some cool tools to check in on the status of the collection in the garden without having maybe staff resources to go out and be checking everything constantly. So since 2015, we have been busy. This is really just a project. I've been the only staff member on this until 2 years ago. I was able to hire a part-time project manager. And otherwise, this has been me reaching out to gardens, asking them if they'd be interested in getting involved in this. But in the 6 years that we've been working, we've collected almost 15,000 herbarium vouchers, over 420 families of vascular plants alone, because bryophytes, let's face it, are extremely rare and difficult to grow in a botanic garden. And over almost 6,000 genera and over 12,000 species. So these are represented now in biobanks where they are preserved, and not all of them are yet accessible you can't go to GGBN and find all of these, but you can find at least half of them. One of the delays is the Excel spreadsheet or whatever database system different gardens use to record their collections may not yet be able to communicate well with the GGBN database. GGBN is very easy to use though. It uses the Darwin Core standard. So this is a standardized data format that biodiversity collections institutions have coalesced on, and it allows databases from all over the world to communicate with each other instantaneously. We've grown from a humble collection program at the US Botanic Garden to 3 in the metropolitan DC area, to now 42 botanic gardens on 6 continents. We established an advisory committee in 2016 at the GGBN World Conference that was held in Berlin at the time, formed sort of what we call a coalition of the willing, some really large engaged botanical institutions that are committed to advancing the frontier of botanical research, and we couldn't do this work without them. Places like Denver Botanic Gardens, Missouri Botanical Garden, New York, Kew, it's really been, I can't say enough good things about gardens. I mean, the people who work there, the people who believe in their mission and their passion is amazing. So the resource cost, you're probably wondering, how much did all that cost? So we created an award starting in 2017 to encourage botanic gardens who were a little bit reluctant. And of course, every garden is understaffed and overworked. So we knew this was a big ask. So we said, well, let's take a small pool of money and offer that to see if that might help. It's only up to $6,000 per award, so it is very modest, but it did really help a lot. We now have funded 25 botanic garden collection programs around the world, and the total cost of the entire collection activities, this doesn't include my postdoctoral fellowship, so that would be added, but the actual resources that have gone into the collection are just under $200,000 for 6 years, which is pretty darn good considering how much we've been able to collect. So this is a little bit of my kind of like pitch to y'all to maybe consider participating, or just to consider vouchering your collection as a habit. There are a few gardens that have done this, but some of them really have done this for a long time. Royal Botanic Gardens Edinburgh has vouchered their living collection for over 100 years. And it's a tradition that they've done. It is a great way to keep track of inflow and outflow of taxa that come into the garden, especially when you first accession a new plant or a new, at least taxon. It's a great way to increase the visibility of your collection, to strengthen connections, of course. They kind of speak for themselves, but my favorite is the outreach and the opportunity to engage with visitors to the garden and tell them about research. Because otherwise, the only place very often a visitor to a garden is going to see research is when they read it on a sign. And that's unfortunate. So to see people actually doing the collection is very fun. Benefits, of course, of membership in GGI, we developed some benefits, including we offer free barcode sequencing and DNA extraction from any new genus or new family that is collected that is not already in GGBN. We include a data embargo on that so that the garden can use that data for whatever purpose they might be interested in. Of course, we offer an internship program that has been annual since 2015. And a number of other resources like best practices, protocols, webinars that we've hosted for a long time. So I moved here 3 years ago. I'm a transplant from Virginia. Never thought I would find myself in Texas, but I love it here. And was excited because Texas has an awesome flora. It's the second most floristically diverse state in the US. Very diverse ecoregions. There are a lot of cool botanical resources in Texas, including 39 states, at least according to BGCI. And so a priority for me, since I got here and into the future, is continuing to develop and foster these connections among Texas botanical gardens, because I think it's an important thing that we can do. And I've seen the example of a lot of the botanical community here already being very supportive of me as a new botanist here. People reach out to me all the time saying, hey, Morgan, just saw, you're a new staff at Brit. Wasn't sure if you've been to this garden or if you'd like to come out to this private land where I know someone and you could collect. It's very cool. So anyway, this is a project that it's a labor of love for sure. And so regardless of whether I continue with GGI Gardens worldwide or not, it's still up in the air for me because it's a lot and I'm getting, well, without a big chunk of money, it's hard to continue. But in Texas here, it's much more manageable. It's a lot of fun. And I love using this as a way to learn about the Texas flora. So, and of course, gardens are kind of the first line of defense for plant conservation. And if that hasn't been apparent through the talk, I hope at some point it becomes so. At BRIT, we have a lot of really cool new resources to help support GGI Gardens activities, but also just generally botanical research in many ways. And so the timing is just right to strike when the iron is hot. We have a new conservation seed laboratory that opened in 2019. We have a new molecular and morphological laboratory with a SEM facility that is available right now for free to use by visiting researchers. We don't have any policy in place right now to charge people. So if that is a lucrative thing, please feel free to come use it. And then we also opened our biorepository in 2019 and joined GGBN as the first botanical biobank as a GGBN partner in Texas. So with all of these resources, we really hope at BRIT to be a leader in botanical research for the state, but to be a resource more than a leader, to provide support for advancing botanical priorities. This is kind of really a thank you slide, but it also just, I think, captures every year when I do my annual report for GGI, I'm kind of blown away. And this year in particular was really special. We got a very generous donation from the US Botanic Garden to fund at least 15 GGI Gardens partner awards. And so we funded 22 in GGI's history since 2017. 15 of those were this year alone, thanks to US Botanic Garden and BGCI. So here's a list. It really helped us internationally increase our representation. And of course, the flora of the United States and the flora that's represented in gardens, there's a limit, right? We don't have everything. So if we ever hope to achieve those high priority goals of GGI for the plant tree of life, we've got to be thinking regionally and internationally. So with that, a lot of people to thank, but especially to the department for welcoming here. It's been a really nice visit. So I'll take any questions. Yeah, Dan.
What's the nature of accidental production of this?
Yes, the question was, what is the danger of accidentally perpetuating misidentifications from botanic gardens into the online databases that are used by researchers? That's a great question. I think anytime you work in a collection, there's that danger. And so there does need to be some steps to verify the identity of these taxa. This is one of the reasons we do DNA barcode sequencing. It's not perfect, and it's certainly not a solution for species level identification in 99% of plants. But at least it can help avoid drastic misidentifications, things that are identified maybe to the wrong family, or even in many cases, the wrong genus. So that's a first line of defense that we build in, but it's not an ultimate solution. We really depend on gardens to have an accurate identification. And the key to preventing this from becoming too pernicious is the voucher. Everything comes down to a voucher. A shocking statistic that I learned just this year, it was published in Science or Nature, is that only 13%, I think, of all vertebrate genomes published have a voucher specimen. So you can imagine we have all these genome sequences, which are incredible resources for us, but we can't confirm the identity of right now 87% of them because there's no voucher. That's a problem. And so that is something that the simple act of making an herbarium voucher can circumvent years of confusion and problems. But I do think it's always going to be a problem for collections-based research. All we can do is our due diligence and give it our best approach. Good question. Yes, Oscar?
Related question. How do you keep up with the taxonomy? How do you keep it straight?
The question is, how do we keep up with the taxonomy and how do we keep it straight? Great question. There are, right now, we have used the plant list as our standard. That's what we started with in 2015. The plant list, of course, is no longer being updated and includes a massive amount of taxonomically unverified names. So we continue to use the now World Flora Online, which has incorporated the plant list taxonomic backbone and has committed to providing curated and up-to-date taxonomic information. And right now, that's the best that we know how to do. World Flora Online, if you don't know, is a project that was started in 2012. It was created as a way to address target one of the global strategy for plant conservation, which is part of the Convention of Biological Diversity. And target one says by 2020, we will have an online digital flora for all species of plants. That goal was not met, but the World Flora Online has at least created the infrastructure for it and a permanent living specialist network. So the World Flora Online is informed by what they call taxonomic expert networks. And these are groups of specialists in different taxa of plants that contribute to this. One that I'm a part of is the International Composite Alliance, which manages a database that feeds into and communicates with the World Flora Online called the Global Composite Database. And yeah, great question. I love that the first two questions immediately highlight the taxonomic impediment because it's serious.
Where do you find the next?
The question is, where do we find the experts? They are self-identified and they are recruited. They're actively recruited and self-identified. In the case of TICA, the International Composite Alliance, we reached out to the World Flora Online. We had a meeting in October 2019 in Belgium. The Global Composite Database is hosted online very generously by a Belgian government program called the LifeWatch Institute, which also manages the World Register of Marine Species. So WORMS, great acronym. They've generously agreed to host the Global Composite Database. It's the first database that is non-animal based. It's the first plant database, and it's really cool. It allows you to link to other databases. So you can link, when you click on a taxon or you search for a taxon, you can link to the original reference, the original publication of that taxon. You can request the publication if it's under copyright from a taxonomic editor, a specialist. You can upload images. You can put taxonomic keys, digital taxonomic keys into their website. It's really cool. I hope that they continue, the LifeWatch Institute, to support other non-marine groups. Yes? That is a great question. because this was a Smithsonian institution funded project, and that's another hidden cost that's not reflected in the actual resource cost. Great question. Oh, sorry, the question was for viewers online, how do we support the funding of sequencing, the barcode sequencing that we do? And Smithsonian was and is the headquarters for, oh gosh, I always confuse these, the Consortium for the Barcode of Life. So there's the International Barcode of Life Project and the Consortium for the Barcode of Life. And so they have funding that supports barcode sequencing for any species that does not already have a barcode sequence available and searchable in the Barcode of Life database, BOLD. Good question. Yes.
Dr. Gostel, I'm not sure if you can hear me.
Yes.
Yes, so I have a question. This is Luis Cisneros. I'm a professor here in the department. And I have been working with different centers of biodiversity, mostly in Latin America. And I've been working with, say, with Peru and the International Potato Center, and also in Brazil with different universities. And one of the big concerns that these centers of biodiversity have is the fact that a lot of these materials should not leave the country because there could be issues of intellectual property, issues that mostly for those big companies that go, like pharmaceutical companies, and they're searching for products and they patent these things. And usually countries become very protective. So I know that there are rules and laws that are right now in place in many of these countries. How do you manage with those issues when it comes to collect materials, and how do you protect them once you bring them? Because one of the issues that I've seen that has happened throughout the years is that a lot of materials that have been transported through programs of collaborations for research purposes mainly went to other countries, and then at a certain point, I'm assuming that the lack of control, these products ended up being transferred into the commercial, activities, and a lot of products that were originally not intended to be commercial ended up being commercial. I'm talking about the purple sweet potatoes, purple potatoes that you find here in the U.S. that originally are from South America and are from the collections that came from the International Potato Center. They ended up being commercial. So how do you deal with these issues? Or this is something that not necessarily is an issue from the things that you've been telling us? Thank you.
Great question. Thank you. Yes, it's a very important issue for everyone who does biological research, and it should be because it's a very valid concern. And so I have a couple of answers to that question that reflect how we have tried to approach and navigate international policy, especially what is most prominent now is the Nagoya Protocol, which governs the equitable access and use of genetic resources, which basically says that you have to do a couple of things. And it's on the collector or the researcher to do their due diligence to ensure they are compliant with the Nagoya Protocol. The Nagoya Protocol is part of the Convention on Biological Diversity. Unfortunately, that has not been ratified by the US. But, and this is an important but, that does not mean the US is not beholden to the terms of the Nagoya Protocol. Biopiracy and bioprospecting are serious problems with potential serious consequences, obviously. And one of the really cool things about GGI and GGBN really is a conscious built-in protection of country rights through the GGBN structure. GGBN partners, in order to be a partner of GGBN, you have to comply with the Nagoya Protocol because you are dealing and handling the sovereign genetic resources of the countries where those genetic resources come from. And there are a couple of things. One, most GGBN biorepositories do not own the rights to use, access, or distribute the material that is in those biorepositories. Some of them are. However, when material enters a GGBN biorepository, there is a material transfer agreement, which is legally binding, which establishes the access and benefits that are expected associated with the use of that specimen. So if those terms of the material transfer agreement are broken or violated, there are legal consequences for that. And this is a standard that GGBN has developed. In a way, it has become a model for other organizations to follow. In a way, it helps GGBN be not an enforcer, but at least a mechanism to prevent circumventing these international policies. Because if GGBN is the leading organization providing these genetic resources, researchers need to go through GGBN, and GGBN is enforcing international policy like this. So that is crucial. It's a huge help. And it has really been helpful establishing international agreements with other countries. Another thing is GGBN is it's not fledgling anymore, but it's a growing organization. And one of the ways they're really trying to grow is by supporting the growth of biorepositories, biobanks that are GGBN partnered in every country on Earth, so that so that countries aren't dependent upon the global north, for example, to deposit and curate and take care of tissues, but we can support and build capacity for collections that are preserved and well maintained in the country where that material originates. And that gives the country obviously complete rights to distribute for research use. And that's, in an ideal world, what we hope to see is every country having their own, whether it's centralized or not, or associated with a private nonprofit, what have you, biobank that can connect researchers to their collections and really, in the future, create like a utopia of harmonious genetic resource use and access. But hopefully that'll happen.
Thank you.
Thanks.